Hello everyone. Today is the last day of my this week's 7-day read an article challenge. I took a 7-day read an article challenge in order to challenge my brain, my mindset about reading scientific articles, and I really feel good about taking this challenge. So by the end of this challenge, what I had hoped to gain was to have more information about the topic of my interest and improving my memory function that is present and helping me to process informations faster. And I liked doing this workout because it actually helped me to read articles much more easily, because I was trying to make this reading of article fun for me by making it into a PowerPoint every day and to go through each of the results and different kind of methodologies that were associated with each of these scientific articles that I was reading throughout these 7 days. So today is the final day of my workout, and for this I have chosen a review article that is talking about the MD simulations that happen during photosynthesis. This talks about the various photosynthetic complexes that are involved during photosynthesis, and how MD simulation kind of helps us to understand the interactions of different factors involved in the photosynthesis. As usual, the link of the paper that I've used for studying will be present in the description box. So without any ado, let's go to the introduction. Basically, these experiments that were discussed in the paper were molecular dynamic simulations that were done on photosynthetic complexes. So as we know, photosynthesis occurs in plant thylakoid chloroplast, and in the thylakoid membrane there are mainly four different proteins that are present that actually help in electron transport during the light reactions that help in the generation of ATP during photosynthesis. These different proteins are the photosystem proteins, uh, photosystem 2 and photosystem 1, the protein cytochrome B6F complex, and the protein ATP synthase that actually helps in the generation of the ATP. Initially, when there is illumination by light, the photosystem 2 can get the electrons and then transfer it to plastoquinones, which then transfer it to cytochrome B6F, that then transfers these electrons via plastocyanin to photosystem 1, and then photosystem 1 can further give out these electrons to reduce NADP to NADPH, and then the transfer of electrons occur that helps in the conversion of ADP to ATP by the ATP synthase due to the difference in the electropotential energy that happens throughout this thylakoid membrane. Now, this electron transfer process can be affected by various different conditions in the environment, or due to different conditions in the pH or the light intensity that could change the overall structural organization of the thylakoid membranes containing these proteins, and also the kind of diffusion amongst these proteins can change under different conditions. And in order to look how these complexes differ in their organization throughout the thylakoid membrane at different time points, molecular dynamic simulations are a good way to study them. So generally, the conformational landscape correlates not only to the protein's energetic landscape, but also to the optical properties that are present for the pigment to protein complexes. Example, like different conformations of light harvesting complexes correlate with different spectral properties that can be observed experimentally. Now, the various states in the photosynthetic systems are separated by different energetic barriers. And if some system has a higher energy barrier, then that system is probably not that much favorable to be present during any time point, rather than a system that is having a lower energy barrier, because in that case the system can change from one form to another form during the time course of exposure to various energetic conditions and be feasible in real life situations. Now, light harvesting complexes have an inbuilt ability to switch reversibly between different states and from more or less quenched state, and therefore have different conformations. LHCs are thus defined as disordered systems with low energy barriers. Now, changing different physiological conditions can change the arrangement of these photosynthetic subunits and change their conformation from one state to another state. And the energetic landscape actually depends or helps in the conformational change in between the different states of the photosynthetic subunits and also in their organization. A lot of molecular dynamic studies have been done with biological systems, and they have been done from a very small amount of time ranges to very high amount of time ranges and in various atomistic resolutions for different systems. Now, studying of bigger biomolecular systems can be really hard because these are very large systems and the time required to study the energetics of the whole system might be more required in order to have a proper sampling of how the conformation of the biological system would look in real life. In order to study the different conformations of the biological systems at different time points during molecular dynamic simulations, there has been development of special kind of parameters called force field, and we are going to discuss about it in a few moments. And the range of these different force parameters that are applied to the different atoms or atomistic situations for each of the different biological samples can have a big role in how much time it requires to do a computation of these systems in the molecular dynamic simulations. On this slide, we have a figure that compares the spatiotemporal scale of MD simulation to the accuracy of the results that are obtained by various different kind of resolutions of MD simulations, such as the quantum mechanical resolution versus the coarse grain resolutions. And what are these? We will discuss in another slide that follows from this slide. Now, as I have told in the previous slide, when the system sizes increases, a bigger simulation time is required in order to figure out the proper visualization of the molecular arrangement in the system that we are investigating. Because now the time has increased, that would cause a decrease in the accuracy of the details of the molecular system that we are visualizing. The initial method by which molecular dynamic simulations can be studied is by doing quantum mechanical or quantum mechanical with molecular mechanics calculations. What are these quantum mechanical and molecular mechanics calculations? These are calculations that consist of electronic and nuclear degrees of freedom for calculation of the movement of the various atoms that are present in a system. This help in a ready characterization of energy transfer and studying the electron transfer process. But this is easy for smaller systems where only a group of atoms are present, but not for bigger systems such as protein molecules. And that's why in the case of protein molecules, bigger kind of resolutions are required to study the larger systems such as the atomistic levels or the coarse grain model systems for molecular dynamic simulation studies are required and have been done successfully. So molecular dynamic simulation is based on the principle of building up a biological system consisting of classical particles mainly atoms or combination of atoms that follow dynamics present in a simulation box. Basically, a simulation box is a box in which we are observing this movement of the various atoms, and the movements of these atoms produce a specific trajectory due to the motion of the atoms. This motion of the atoms is due to the velocity of the atoms, which are based on the coordinates of the atoms from the initial file system, such as a protein database file coordinate system, or from any kind of coordinate system that has been studied by experiment, and the velocities that could be present for each of these atoms when a specific force is happening on these atoms to move these atoms from their initial position to a final position by a displacement due to a specific velocity. Now, the forces that act on these atoms are computed over time, and they are dependent on the particle positions and the way the particles are oriented with respect to each other. And for that, a few parameters need to be known, such as the distance between two atoms in a system, the angles formed between various atoms in a system, and the dihedral angle of these different atoms that are present in two planes of a system. From these different parameters, the bonded forces and the non-bonded forces can be calculated, and they sum total to the final potential energy that gives the potential energy required by this different set of atoms to move from an initial position to a final position that we see during the molecular dynamics simulation. So in this figure over here, we basically see a beta carotene molecule, and we also see the various parameters required by this beta carotene molecule to finally calculate the total potential energy of the system that will give us an idea of how much energy was required for a final displacement of the total molecule. Now, in order to see the whole information about this system, there has to be specific force field parameters that are associated with each of these atoms. These force fields are calculated based on the potential energy terms that were developed from the forces acting on each of these atoms. And force field parameters could be of two types: the atomistic force field type or the coarse-grained force field type. So an all-atomistic model, called also all-atom force field, actually consists of the model where all of the atoms of the system are considered in solving the equation of the motion of the atoms involved in the system. So in the figure B, you see the different levels of resolution that can be seen by molecular dynamic simulations based on different kind of force fields that are applied. So the all atom force field is denoted by the AA force field image over here that considers all of the atoms present in the beta carotene moiety and all of the forces that are present in each of these atoms for the movement of these atoms to take place over time. The second model of the level of resolution that we are going to discuss consists of force fields that are known as united atom force fields or UA force fields. In future, we will be referring to as united atoms always as UA. And what this does is that it consists of all of the force fields from atoms that are not hydrogen, but in certain atoms where the hydrogens are associated to nonpolar groups. In that case, those hydrogens are not going to be considered as separate moieties with respect to the hydrogen atom as particle, and they will be just considered, their forces will be just considered implicitly. So the exact forces for these hydrogen atoms that are attached to the nonpolar moieties will not be considered directly. So this actually helps in uh, fastening of the process of molecular dynamics simulation, and that's why important in certain kind of MD simulations where we still need or want most of the energetic information that we can get in an atomistic force field model, but we need it faster compared to what would have been generated by just an all atom force field kind of simulation. Now the coarse grain force field that is generated, that kind of force field actually uh, calculates almost all of the atoms that are present in the system, not individually but as clustered group of moieties. So there would be 3 to 6 non-hydrogen atoms that are considered to be single groups within the moieties, and they will form a specific kind of beads for the coarse grain force field, on which the forces will be applied, leading to the motion of the overall bead present in the coarse grain. And this force that is now being applied on these specific beads of the coarse grain are actually forces developed due to the summation of the forces that would have been applied for each of those specific atoms, but it's not a direct summation, so there are certain parameterization that is done on the forces that will be applied to the coarse grain, and this makes the calculation for the MD simulation faster. In order to generate the coarse grain force field, there are two different approaches that have been used. One is a bottom-up approach that is targeted only to the structural properties of the coarse grain model, and the other one is a top-down model that is developed to target the experimental ensemble of the coarse grain model that is present, and that helps in tackling of the force field parameterization that is present for the coarse grain modeling of MD simulation. In larger biomolecular systems, most of the times coarse grain models are favored because they make the simulation time faster and helping in larger microsecond time scale of simulations that can be done for biomolecular systems. Now let's discuss about the workflow for a molecular dynamic simulation. Initially, for the molecular dynamic simulation, there is the necessity of building up a simulation box. So this simulation box consists of the protein molecule ensemble that is generated from the proper PDB structure of the protein that are involved in the protein complexes, generally from a PDB data bank or a protein database bank. And these molecules will be arranged along with the proper force field parameters that are required for the MD simulation to occur. Remember, each of the biomolecules have different atoms or groups of atoms present at speeds, and on each of these speeds a specific set of forces are required in order for the displacement of these atoms or the beads to finally see the uh, dif difference in the initial versus the final coordinates of these atoms or the beads due to molecular displacement that will happen over time due to molecular dynamic simulations, and that would give us a final realization or understanding about how different atoms or molecules move around, changing their conformation or organization over a period of time. So once the simulation box is made, the next step is to minimize the energy, and the energy minimization is required in order to properly understand or make the effect of non-bonding interactions that are present in the molecular interactions proper, because if there is not proper interactions that are being uh, developed in the molecular dynamic simulations over a period of time, then the energy that will be produced uh, after the whole MD simulation process is over could be very high, to an extent that these atoms won't be converted in a state in a specific conformation, and the higher energy could lead to disrupture of bonds that are not suitable to visualize the molecular system in its native condition. The third step is to apply a periodic boundary condition, and that would be done to assign proper cutoffs to the potential that are present, so that the energy of the system is properly regulated under specific conditions. And these conditions that we are talking about are more important in the equilibration state, where we want this conditions that are applied on the molecular ensemble, such as conditions of proper pressure and temperature, or proper volume and temperature, are kept at a specific condition set for the molecular dynamics to happen in such a way that there is the energy of the molecules will not exceed to an extent that would blow up the whole solution, whole molecular system, but would help to visualize the molecules with minimized perturbations. And on completion of the equilibration, the final system can be visualized by visualizing the root mean square deviation of the atoms from their initial position, in order to see if there are certain atoms or bits that have moved out to such an extent that they are not anymore confined within a specific system bounded by NPT or NVT conditions. And the next step is to do a production run, where these different trajectory movements that happened over time are done at longer time scales and with independent replications of this molecular dynamic simulations are done. And after the production run is done, then each of these trajectories are analyzed in order to figure out that what kind of conformational changes happened with respect to different times throughout the time scale, and if there was some kind of energy barrier that was observed by the molecule and it really didn't go into its lowest energetic state in order to have the proper orientation or not. Now, this whole process of the MD simulation needs a specific amount of time that is required for the simulation to give a final orientation or organization of the biomolecular complex in such a way that it represents the native state. And for that, there could be necessity of higher microseconds of simulation that is required. But in order for these simulations to happen uh, at a larger time scale, it is important that the uh, force fields are parameterized in such a way that the forces associated with this motion of the particles can be properly analyzed, and the forces are calculated as the negative derivative of the potential energy that is changing during the MD simulation, and depends on the force field parameters and on the position of these atoms. In order for this derivative to be calculated, there are various numerical integration methods that are used in order to calculate the equation of the motion of the atoms, such as the Euler integrator or the Verlet integrator that could be used for the time evolution of these atoms to be visualized. In current days, the MD simulation studies that are being done are using Verlet integrator because it helps in uh, estimating the different conformations at a faster time scale than the Euler integrator method. Due to different energetic barriers that can be faced by the atoms present in a molecule, sometimes the samples, when they are run through MD simulation, do not result in the final orientation of the molecule that would be expected, and even after sampling of a huge number of trajectories, the proper results might not be obtained. And in order to get rid of such of lack of sampling that is observed during MD simulation, there could be two different methods that could be used. First is to run multiple independent simulations with random velocities at each time. This process can be done, but it is highly time consuming, and it might not still end up giving a appropriate result. So there is a second method that is often used, and it is known as enhanced sampling. Now, enhanced sampling can be of two types. One in which there is a predefined reaction coordinate along which the sampling is being enhanced, and the second one is where there is no specific reaction coordinate against which the energetic profile of the molecule is being studied. Now, one of such method of enhanced sampling is the umbrella sampling method that belongs to the predefined reaction coordinate system of enhanced sampling, in which, in absence of a bias, we would see a specific energetic profile of the energy versus reaction coordinate for the molecular system, as you can see in figure B. The solid line represents the original profile for the molecule with a high energy barrier, and because of the high energy barrier, it is not being able to properly sample the exact conformation of the molecule. But if on this potential there is application of additional biasing potential, that would lower the energetic barrier that is present during the sampling of the molecule and allow a faster sampling of the molecule over the different time scales, and also show the proper lower energetic conformation of the molecule that would be present in the original um, in vivo studies that would be expected from experimental studies. So, along with umbrella sampling, another kind of predefined reaction coordinate sampling is the steered molecular dynamics study, which is shown in figure A. And if you see over here, this molecular structure is that of beta carotene inside the membrane, and in order to do steered molecular dynamics, what is done is an external force is applied in such a way that now one of the systems, or one of the systems of combination of atoms, would start to steer out from the rest of the system, as you can see by the black dashed line that actually shows the force in which the external force is being applied, so that this beta carotene moiety can now steer out from the rest of the complex in such a manner that the overall energy barrier for this moiety is decreased, and so now since the energetic barrier has been decreased, the molecule can now sample a different region in the potential energy profile in order to have a lower energy state at which this sample is much more stable. The third way of doing a predefined reaction coordinate is metadynamics, and in this system what it does is that it has a history dependent biasing potential that is based on each of the trajectories on which biasing potentials are added, and eventually all of these biasing potentials are cumulated together, and they are also known as collective variables that are applied on these systems that could lead to a change in the energetic barrier of the sample with time, lowering the energy of the system and uh, giving it a energy profile where the in the molecule ensemble can be present in a proper low energy state. The metadynamics is shown in figure C. Now, amongst the kind of enhanced sampling that do not require the reaction coordinate is accelerated molecular dynamics, in which there is a time-independent bias potential that is provided, which is generally added if the biasing potential that was present for the initial potential energy surface that we see is lesser than the threshold value. And the second way that does not require reaction coordinate is a temperature replica exchange method, as you can see in figure E, where if we change the temperature from one temperature to other, it would help at the change of the molecular flexibility of the molecular ensemble, and thus help in the molecule to attain a orientation at which the molecule has a lower energy that fits its proper orientation in the original structure. The interplay between the pigments and the protein complexes that are present in lipid membrane have been studied using molecular dynamics, using both coarse grain model and even atomistic models in certain cases. And this paper provides a high background on the different kind of experiments that were done in order to visualize different kind of interactions. I'm not going to go through all of them, but I would suggest you to read this paper in order to see how there has been interactions of different lipid membranes that have affected the different amount of interactions of the different kind of uh, pigments such as beta carotenes, or even the diffusion of the uh, pigments around the PS2s in different kind of lipid environments that are present. Plastoquinone, which plays a major role in electron transfer, that has been studied using MD simulations as well, and the plastoquinone's diffusion through the photosystem 2 has been studied in order to figure out the channels of exchange in which the plastoquinone can reorient themselves in a flip-flop manner. Although flip-flop for the oxidized plastoquinone was seen, but not for the reduced plastoquinone was observed for the time scale of the molecular dynamic simulation that was done. This kind of studies actually show the interactions of smaller molecules such as plastoquinones to that of photosystems that are present during photosynthesis. Lipid binding sites of photosystem 2 were also analyzed, and this paper actually shows that how, due to the different charges of the different kind of lipid moieties, there could be different kind of interactions that are present around the light harvesting complexes, and because of which there could be different kind of orientations of the lipids surrounding the different kind of photosystems that is possible. This paper also talks about the flexibility of the proteins that are present or exposed to the membrane water environment versus the proteins that, or the regions of the proteins that are involved in the core protein subunit that are present in the light harvesting complexes. So overall, this gives an idea of how the environment surrounding the proteins can actually orient the different regions in the protein complexes that are present. This paper also talks about the different disorders that has been seen with the carotenoid sites that are present in the light harvesting complexes, and it also talks about how the effect of pH can lead to the change of the conformational dynamics that are associated with light harvesting complexes. I'm not going to discuss a lot about this paper because I think it is much easier to read this paper, and it is better to understand each of the different results that were observed by the different procedures of MD simulation that were done uh, individually rather being discussed over here. With this, I conclude the 7 days of reading an article challenge that I had taken up. So far, I had a very good experience with this challenge, and I really enjoyed reading these papers. This last paper that I read, although it was a review article, I still really enjoyed it because it taught me a lot of different kind of parameterization that goes into the playing of molecular dynamic simulations that are associated with photosynthesis, and how these parameterizations can finally affect the results that are being obtained, and how these results can vary or also concur with the experimental results that have been already found out. Uh, if you are interested in such kind of topics, I would suggest that you can read this article. So far, in order to keep me motivated in reading an article, I had created this chart through which I would click on every day, for which I would read the article, and this has helped me in keeping motivated to read an article. You can do a similar kind of thing. If you want to take up a 7 day challenge, I would suggest you to definitely take up a 7 day challenge of reading an article. And if you like such an challenge, then please put the thumbs up link that is present under your video screen, and also subscribe to my channel. I really enjoyed, honestly, this reading of 7 scientific articles throughout 7 different days of the week, and I encourage you to take up this challenge. Thank you for watching my video, and have a good day. Bye.